Hello, science fans, and welcome back to G Fun Facts Online, the show that dives deep into the coolest science stories out there. Today, we're talking about something truly mind-blowing, a real-life Noah's Ark, but instead of a giant boat, it's a super-chilled vault. We're exploring the fascinating world of cryopreservation ecology, basically, freezing life to save it. Now, we all know our planet's amazing biodiversity is in a tough spot. Climate change, habitat destruction, it's a rough time for countless species. Wildlife populations have plummeted by an average of nearly 70% since 1970. That's a staggering loss. But scientists are fighting back with some seriously cool tech. Literally. Cryopreservation is like hitting the pause button on extinction, creating a biological safety net to protect life for future generations. It's not a magic fix, but it's a vital tool in our conservation toolkit. So, how do you freeze life without, destroying it? At its heart, cryopreservation means cooling living cells, tissues, and other biological bits down to incredibly low temperatures, usually in liquid nitrogen at a frosty -196 degrees Celsius or -320 Fahrenheit. At these temperatures, all the biological action inside cells just stops, allowing for super long-term storage without them degrading. And what exactly are we putting in this frozen ark? It's a whole menagerie. We're talking reproductive cells like sperm, eggs, and embryos, which are key for future breeding programs. Somatic cells, like skin cells, and even whole DNA are being banked, offering a treasure trove of genetic info for research, and maybe even future cloning. For plants, it's seeds, especially those tricky recalcitrant seeds that don't like conventional storage, plus pollen, shoot tips, and cultured cells. Groundbreaking work is even being done to cryopreserve delicate coral larvae and fragments. And let's not forget the tiny titans, microbial cultures, which are crucial for healthy ecosystems. The biggest challenge? Ice crystals. If water inside cells freezes into sharp crystals, it's game over for the cell. To dodge this, scientists use a few clever tricks. There's slow freezing, where samples are cooled gradually with cryoprotective agents, or CPAs, that help draw water out of cells before it freezes. Then there's vitrification, which is super fast cooling that turns the water into a glass-like state, no crystals allowed. Plants have their own special techniques, like encapsulation dehydration. And for complex things like coral fragments, there's isochoric vitrification, which freezes at constant volume to prevent ice damage. Those CPAs, like glycerol or DMSO, are crucial. They lower water's freezing point and make it thicker, stopping ice. But they can be toxic themselves, so finding the perfect, gentle CPA recipe is an ongoing quest. Let's look at where this deep freeze tech is making a difference. For animals, think